Good afternoon. I'm Larry Marnett, the Dean of the School of Medicine Basic Sciences, and I'm very pleased to welcome you all here today for Basic Science Dean's Lecture by Dr. Barney Graham. Barney has been chosen the Distinguished Alumnus Award winner for 2021 by the Vanderbilt Alumni Association. He is truly an inspired choice. He is a path-breaking immunologist, virologist, educator, and leader, and was the chief architect for the first experimental COVID-19 vaccines at the National Institutes of Health, building on decades of research in his laboratory. Dr. Graham grew up on a farm in Kansas and went to Rice University, where he majored in biology. He finished his medical degree at the University of Kansas and came to Vanderbilt University for internship and residency. He served as the chief resident at Nashville General Hospital and head of the Vanderbilt AIDS Vaccine Evaluation Unit, where he led the first human trial on an AIDS vaccine. While in these roles, Dr. Graham completed a PhD in microbiology and immunology at Vanderbilt. In 2000, he was recruited to the NIH to create the Vaccine Research Center, where he stayed until his retirement earlier this year. Barney has been a seminal contributor to the design and development of vaccines against respiratory pathogens, including SARS-CoV-2. His studies of the spike proteins enabled customization of mRNA vaccines to produce the most effective immune response. Barney saved millions of lives through his work at the NIH Vaccine Research Center. He has been recognized with many awards, including election to the National Academy of Sciences earlier this year. It is a tremendous honor to welcome him back to Vanderbilt for today's lecture and to receive the Distinguished Alumnus Award, which will happen during the reception that follows. One other note I'd like to make is that Barney is a tremendous mentor, generous with both praise, credit, and with his results, which has expanded the impact of his science dramatically. Today, his lecture is entitled Reflections on Pandemics and the Future of Medicine. Please join me in welcoming Barney to the podium.
Well, it is a real pleasure to be home again. Lots of memories flood back when I'm in this place with all these people that I know so well. And many of you came back today unexpectedly to see me, and so thank you. And I'm going to review a little bit about what we've done at the VRC over time, but try to put it in the context of what I think I learned at Vanderbilt and about being a doctor. So my thoughts right now are going back to the office where Debbie worked and let me interview on Christmas Eve to come to the residency program at Vanderbilt without notice. And at that time, Dr. Bittingham and Dr. Little and the chief resident were all in their offices working that day on Christmas Eve, and all interviewed me and allowed me to come here. So I'm got a lot of thoughts in my head right now. I'll try to get started. So I've got some disclosures on vaccine and monoclonal antibody patents for different virus diseases. And I left Vanderbilt to go to this unique place called the Vaccine Research Center that was founded to develop an HIV vaccine. And this is our basic research building, Building 40 on the NIH campus in Bethesda. And it's unique because it's a very multidisciplinary group that founded this. And it not only includes basic research, but process development, pilot plant manufacturing, a self-sustained contained clinical trials unit, and a laboratory that analyzes the clinical samples. And it's allowed us to do more than just HIV over time to to look into some of these unmet needs and to respond to a number of emerging infectious diseases using a whole variety of different modalities, including nucleic acid vaccines and vector-based approaches, VLPs, proteins, including nanoparticle displays, and monoclonal antibodies. And these have been shared around the world, and it's been done by a relatively small group of principal investigators and program heads, some of whom are still there and some of whom have already left. And so the VRC is in the process of going into its next era. And I gave myself 5 minutes to write down a few names that just popped into my head, and I can't really talk about everybody, but I could give a whole afternoon talk about the stories from the people's names who are on this screen. I don't have time to do that, but I want to thank everybody here, plus everybody who's not listed that probably ought to be here. And I'll just mention Dr. Collins because I'm going to mention some other Vanderbilt alumni and in a few minutes, and also Dr. Spickard, who I'll mention in a few minutes. So I witnessed the beginning of my first pandemic when I was chief resident at General Hospital. And this is located, the building may not even be there anymore, but you might be able to tell where it is because the Titans Stadium is over on this side of the river. General Hospital was there. It's now located on the Meharry campus, but this is where I was chief resident in 1982. Dr. George Thiel was my chief of medicine, and shortly into my chief residency, a man came into the screening clinic and into the emergency room and had a condition that nobody really understood very well. He was sent out. I put a note in the chart to have them call me if he ever came back. And he did a few days later, and he had all these conditions all at the same time. And he had cryptococcus in his spinal fluid that pelleted before you finished the LP. And he had so much Mycobacterium, avium intercellulare, in his spleen and liver that you could see it on a blood smear, let alone in the spleen. And he had pneumocystis and CMV and everything. And at that time, only syphilis really caused everything to happen in a person. And at that time in medicine, the idea was that there should only be one cause of disease. And so this was extremely confusing case. And we didn't really measure CD4 T cells at that time, but we got that done, and he had no CD4 T cells. And he was the first, or one of the first patients with AIDS in Tennessee. And that's what started, I think, my course toward being a virologist. So internists at that time usually didn't do viruses, not very often. And so the pediatricians were the ones doing virology. And I went to Peter Wright and David Carzon, and you see Kathy Edwards there, the pediatricians doing virology, and I told them I wanted to be a virologist. And so how do I do that? And Peter said, well, start by growing respiratory syncytial virus. So I did that. I spent about 6 months just learning how to grow the virus in cell culture. And Edie Sinella, who's here in the audience, helped me learn how to do plaque assays and 12-well plates and the whole thing. But we learned how to infect mice. We studied the immunology. Vanderbilt let me open up a broom closet next to my office and put this old incubator inside so that I could keep the mice right next to my office because I didn't want them contaminated with other things from the animal care. So I'm grateful for that. And we defined RSV. Since then, I've experienced a number of other viral threats and emerging infectious diseases over time, and I'm listing some of them here. Still, the biggest one is HIV, which was basically a regional emerging infectious disease that wasn't recognized until it became a pandemic. And we're going to get back to that idea at the end. So these are some of the deadliest pandemics in history, represented by size. Many of them were based on a bacterial disease, Yersinia pestis or the plague. And some of them were based on systemic viral infections like HIV or smallpox, shown here. And then many of the other ones have been respiratory viruses, including the 1918 flu pandemic, and then some of these, which we used to think of significant flu pandemics until coronavirus came, and now I'm representing it as something that's probably killed closer to 20 million people on Earth, and without the rapid interventions may have killed twice that many. So if you look at those pandemics, and I'm generalizing here, but many of the pandemics were preceded by wars and social conflicts, things like that, that we really created ourselves. During the pandemics, there was a lot of misplaced blame, increasing political polarization, racism, all sorts of things. And we've witnessed that even during this last couple of years. But after these pandemics, there was often a period of relative enlightenment where many times people who were older, who had been making all the decisions and leading everything, things shifted to a younger set of decision makers. We developed greater diversity and mixing of cultures. There were innovations in technology and governance and art, less economic disparity and rise of middle classes, and more interest in compassion and justice. So the question is, as I go through this next part of the talk, is what's going to happen after this pandemic? So in 1918, this is an example of what I was talking about. This was preceded by World War I and a lot of increasing economic disparity caused by the monopolies that were eventually disrupted. But this is what led into the 1918 pandemic. But after the pandemic, we had the Flexner Report, which resulted in the building of the old Vanderbilt Medical Center North Hospital in 1925. The Rockefeller Foundation initiated global programs for virus discovery. They started a unit in Entebbe, Uganda. At the time it was called the East African Virology Research Institute. Now it's Ugandan Virology Research Institute. This is a place that's discovered dozens of viruses, and many of them are now relevant because they discovered chikungunya, West Nile, and Zika, which have all plagued us over the last 20 years, but they discovered those back in the 1940s. They also, it was the home of Oswald Avery, and Oswald Avery was sure that pneumococcal disease caused the 1918 flu pandemic, and he studied the components of pneumococcus. He separated the proteins from the carbohydrates, from the nucleic acid, and tried to assign a value to each one of those components, and eventually found that the transforming principle, the thing that turned smooth colonies into rough pneumococcal colonies, was the nucleic acid. Everyone thought before that it would be the protein. So he was the one in that group who showed that DNA was basically the basis of our genomes. And it was also a time when people wanted to talk about idealism in medicine. And so Sinclair Lewis wrote the book Aerosmith. And for all the young people in the room, you should read Aerosmith. It's about how to be more excited about medicine and research and discovery. And another man who was a contemporary wrote the book Microbe Hunters. And this book is something Anderson Spickard gave me as a going away gift from Vanderbilt. It's a first edition book of Microbe Hunters by Paul de Kruif. And these two were friends. Paul de Kruif had left Rockefeller from some controversy and became a science writer. And Sinclair Lewis was a writer of fiction. So Paul de Kruif wrote this series of biographical stories that were very personal and very much about the personality of the people as much as the discoveries of the people, going through everything from Leeuwenhoek's discovery of the microscope through Paul Ehrlich's random discovery of salvaracin for syphilis treatment. So that is a book also worth reading. That was published in 1926, and Aerosmith was published in 1925. And I think you'll find them both extremely relevant today. The other thing I always wonder about 1918 flu pandemic is this right here, which is a gravestone in Pratt, Kansas. And this was my grandfather's first wife. My grandfather's first wife died in the 1918 flu pandemic, and he didn't marry my grandmother until a few years later. So you wonder, should we be working on these pandemics or not? So the rest of this is going to be a story about how technology has changed the way we think about virology and what it's going to do for vaccinology going into the future. And most vaccines, antiviral vaccines, have been discovered opportunistically based on the availability of new technologies. These first two, the smallpox from Jenner and Pasteur's rabies vaccine, were done before viruses had been discovered, before immunology had been discovered, and they were basically just by the will of individuals growing vaccines on the hides of calves and donkeys and spinal cords of dogs and rabbits. But these next two also relate back to Vanderbilt because Goodpasture learned how to grow viruses in eggs, and that allowed yellow fever and influenza vaccines to be developed. Enders et al. Developed cell culture, and that allowed these live attenuated vaccines and whole inactivated vaccines to be developed. And then the era of molecular biology that gave us recombinant proteins and reassortant viruses and molecular clones and virus-like particles and gene-based antigen delivery, which was used for the recent VSV Ebola vaccine. But I think that the next era of vaccinology is going to be based on a structure. It's going to be structure-based design, this era of structure-based design. And we thought it might be, this picture got mixed up. But anyway, that's supposed to be an RSV-F protein. We thought it was going to be a respiratory syncytial virus that was going to lead the way for structure-based vaccine design, but it turned out that coronavirus came in ahead of that and used not just structure, but all of these other new technologies that have been developed over the last 15 or so years to respond to this pandemic. Some of these technologies have improved the precision of antigen design, or the way we measure immune responses, or the way we, and then the other ones have enabled more speed in response. So this is basically what you need for pandemic preparedness or pandemic response. When I started my career, the way we thought about RSV-F was this blob on a Western blot. And now we can see it in atomic level resolution down to 2 to 3 angstroms. When you wanted to get a sequence of RNA, you had to pour probably 3 times a gel the size of a door and run it over 6 months and hope for the best, and maybe you can get a few thousand nucleotide sequences. And now, boy, these slides have really rearranged the pictures. But now we can generate sequence for 30,000 nucleotide sequences in just a day. We started with serology, just color changes on a 96-well plate, and now we can take polyclonal serum and map it almost immediately, even by antigenic site, with electron microscopy techniques developed by Andrew Ward at Scripps. We also have more precise protein engineering and ability to all my pictures on different slides have been rearranged into different slides. I don't know what's happening. But anyway, this was supposed to be a vial of the Lilly, of the Pfizer product in 1965 that was used in the trial that caused vaccine-enhanced disease. And you can see some brown fluid in the bottom of it. And now we can design proteins based on nanoparticle display with incredible precision. And well, here's the vial down here. And we can make, instead of making fusions from spleen cells, we can generate thousands of sequences from individually sorted B cells in just a matter of a few weeks. Do you think it'd be possible to reload the file? Because these slides are not what I intended them to be. So let me move it onto the desktop. When I was a chief resident, we made our own slides, and they were 2x2 that we showed on a slide projector, and we would go in and use the X-ray machine to develop the film that we had just used to take pictures on a piece of paper. So this is all quite new and different. Hopefully this will work this time around. Nope. Let me try one more thing, sorry. Okay, if it doesn't work this time, we're just going to keep going. Oh, boy. I'm not quite sure what to do, because this is really mixed up. It's not worth showing you the slides. Let me try that. Isabel, yeah. There we go. you know how to set it up?
Do you have an HDMI? Let's see. It might just be this. So if we unplug this, unplug that, sorry, and then plug this HDMI into that side. There we go.
All right. Sorry about that. Okay. That's what it's supposed to be. Okay, so this is how it started, and this is how it's going. That's the bottom line. So it all started for me with respiratory syncytial virus. When I started that work in the pediatric department here in 1985, that was the thing I had a mission to solve. And RSV is an infection, a respiratory virus that infects all of us by the age of 2 and repeatedly throughout life. It infects the small airways, the ciliated cells of the small airways, and the type 1 pneumocytes in the lung, and causes most severe disease in the youngest infants and the oldest frail elderly. And we're going to focus on this protein that's on the surface, one of 3 surface proteins called the F protein for fusion. And we were able to capture the conformation of this protein using an antibody, a new antibody that could hold the top together, and a trimerization domain to hold the C-terminus together. And you see how different this protein looks than the protein that we'd been using for several decades before. And once we captured this protein, it was apparent that there was a site at the apex that was very neutralization sensitive, because the antibodies to this site were now 100 to 1,000 times more potent than antibodies to other parts of the virus. And that site, that red site, didn't exist on the rearranged post-fusion protein, and that had been used in 5 phase 3 efficacy trials over the last several decades, always with the same result, that it would boost neutralizing activity by 2 to 3-fold. But if you stabilized it with disulfides and internal cavity filling mutations and kept the trimerization domain and held it in that shape, it now is a vaccine antigen that can boost neutralizing activity by upwards of 16-fold. And just so you understand, this is why I had to change the slides. This is how these fusion proteins work. The fusion protein exists on the surface of the virus. It unravels, injects into the host cell, pulls it together through these heptad repeat domains, creates a fusion pore so the virus nucleic acid can enter the cell and start the replication process. If you attack that protein, you're attacking the used up, non-functional, spent protein. So you have to attack the original version of the protein to really impact this important viral entry process. That seems simple, but finding that these pre-fusion molecules made better vaccine antigens has changed everything. And these kinds of proteins are on the surface of a lot of the envelope viruses we're familiar with, not just respiratory syncytial virus, but paramyxal viruses, influenza, HIV, Lassa, Ebola, all have class 1 fusion proteins, and some of them are capped with a domain that has to come off for the machinery to work. Coronavirus has not only a cap, but this intervening piece, so it requires 2 cleavage sites to reveal this fusion machinery that mediates this membrane fusion and viral entry to the cell. So knowing this, and knowing that this kind of idea could be generalized, and knowing that we'd gone through these events with Ebola and chikungunya and MERS coronavirus and then Zika, we thought we needed to have a more proactive approach to pandemic preparedness. And the idea was to look at the 27 viral families that infect humans and organize them into entry mechanisms, class 1, 2, or 3 proteins, and then non-envelope viruses, and develop programs where you could learn everything you needed to know about structure, about replication processes, tropism, etc., and use pathogen-specific investigators to study these groups, working together across viral families, all supported by groups that had these capacities, which is what basically we can do at the Vaccine Research Center. So this would be the research part, and this would be the development capacity. And the idea would be to find 30 or so prototypic viruses that would represent these viral families that you would develop products for all the way through phase 1, and develop products at least through animal testing for the other 90 or so viruses that were thought to have human-to-human transmission potential. And so this was our idea for prototype pathogen approach to pandemic preparedness. And we wanted to do a demonstration project looking at both the paramyxoviruses and coronaviruses using Nipah as the prototype here and MERS coronavirus as the prototype here. And because we had just finished the Zika outbreak response with a DNA vaccine made by the VRC that went into people within 100 days, and it was in phase 2B efficacy in Central and South America within about 16 months, but it missed the peak of the pandemic and the outbreak, and we didn't get an answer. But during that year, we also worked with 3 different mRNA companies, including Moderna, and compared that approach to nucleic acid vaccination to DNA. And because mRNA is like DNA, it gives you authentic antigen presentation. It can induce not only antibodies, but CD8 T cells. It only requires entry into the cytoplasm, unlike DNA, which has to get all the way into the nucleus. And so this mRNA was about way more potent than the DNA. 4 milligrams of DNA was like 30 micrograms of RNA for Zika, using almost exactly the same construct. So we made a deal with Moderna to work on paramyxoviruses and coronaviruses. The paramyxovirus approach included first showing that the F protein on paramyxoviruses was a better vaccine antigen in the pre-fusion conformation, and it was. But in paramyxoviruses, you don't know if the attachment protein or the fusion protein is the best target. So we designed these chimeric molecules in which the pre-fusion form was attached to 3 attachment proteins on the other end. So now you have an FG hybrid molecule that can be delivered either as a protein or as an mRNA and effectively protect against lethal challenge with Nipah. We also were doing this for these other paramyxoviruses. For the coronaviruses, as Jason McClellan, who was in Peter Kwong's lab when we worked on the RSV-F protein, as he went to Dartmouth to start his new faculty position, we wanted to extend the finding to other envelope viruses. And so we chose coronaviruses because nothing was known about spike structure. And we were still suffering through the MERS scare in the Middle East. And after a couple of years and quite a few serendipity type events, we finally got the structure of a human beta coronavirus spike protein, which was an endemic virus, the HKU1 virus, and then found ways of stabilizing that fusion machinery, in this case using proline substitutions at the top of the central helix to prevent that pivoting that the molecule has to do to rearrange. And the important thing about this stabilizing mutation was that it not only maintained these neutralization-sensitive epitopes at the apex of this molecule, but when you transduced a cell, it increased expression levels of protein by upwards of 50-fold. So this is MERS spike expression down here without the mutation, and this is MERS expression with the mutation. So for the young people in the room, that work was published in 2017 in PNAS. That was the 6th journal we submitted that work to because nobody really thought it was all that interesting. So don't give up on your papers, just keep pressing forward. The experts don't really always know what's right. So we had this prototype pathogen idea. We'd worked with mRNA companies and wanted to use RNA as the rapid response element and this precision antigen design with using structure as the other element. And Dr. Fauci agreed to this, and we made a deal with Moderna. In September of 2019, we pulled the trigger and said we're going to test the NEPA product in the first quarter of 2020. Moderna was manufacturing the mRNA, and we went to visit and do diligence at the facility and made the decision to make this small batch of RNA and do our NEPA study. And then on January 6th, when we learned that this new outbreak in China was probably a beta coronavirus, I had a talk with Stefan Boncel at Moderna, and we said we might as well flip this demonstration project to our coronavirus instead of the Nipah virus, because we'd shown for both constructs that it could protect against lethal challenge with that pathogen. And so because of this work we had done on structure and mapping epitopes and understanding neutralization mechanisms, and because of the prior collaboration we had with Moderna, and because of the work we'd already done with Jason McClellan, who's now at UT Austin, we were able to order sequences on the 10th of January as soon as they were posted that allowed us to make protein for solving structures, for making diagnostic assays, immunizing mice, and using the sequence for Moderna to give us back phase 1 clinical trial material that started about 65 days after the sequence was released. And this was informed by the RSV work. It was also informed by our thinking about platform technologies and pandemic preparedness, and also motivated because we had been late to the party for both Ebola in 2014 and Zika in 2016. Now, the great thing about this new technology, as Dr. Crow can attest to here, is that you use the same reagents, the same material, the same way of thinking to make not just preventive vaccines, but therapeutic monoclonal antibodies. So as soon as we had PBMCs from a convalescent donor and had the new protein probes for the new spike, we went to our collaborator at AbCellera, who we'd been working with for 3 years on cross-reactive coronavirus antibodies already. They sent us back sequences within about 2 weeks. We made the antibodies, tested them for neutralization, and within a few months, Lilly had turned the better products into bamlanivimab, which was one of the first therapeutic monoclonal antibodies that was shown to reduce illness from people with coronavirus. And from that same subject, the bebtelovimab, which is the antibody that still is effective against the new Omicron variants for therapy, that same antibody, our 1404 antibody, turned into their bebtelovimab antibody. So the same things that can make prevention, the biological reagents and things, can make also therapeutic products. So when coronavirus came, we were in a very unusual position where we had done all this work on technology development and thinking about vaccines. We had a lot of information about RSV and the vaccine-enhanced disease that had happened before that was relevant for coronavirus, because some of the things that happened in the first SARS epidemic were concerning. And we'd been responding to these public health emergencies of international concern. We also had this experience with structure-based design and antibody discovery and the platform manufacturing and pandemic preparedness, and importantly, the pre-existing public-private partnerships, the one with Moderna in particular, and the academic partnerships, because all along for these last 8 years, we've been working with Mark Dennison's lab here at Vanderbilt, and with Ralph Barrick's lab at UNC, and with Andrew Ward's lab at Scripps, and with Jason at UT Austin. So the entire system was there waiting for this kind of response. And So some people have been concerned, and it's been a cause of vaccine hesitancy, that this was done too quickly. And you can tell it as a one-year story, as we just did, but you could also tell it as a 3-year story where we were doing the pandemic preparedness planning, or an 8-year story where we've been doing structure-based design on class 1 fusion proteins, or a 15 or 20 or 40-year story, because it really starts with HIV and the attempts to make an HIV vaccine that led to these new technology developments, not just by the VRC, but by a lot of different groups. And this iterative approach in building little by little the technologies needed to have this type of response is what led to this one-year story. So for us, everything starts with knowing what the protein looks like. If it looks right, if it's in the right conformation, it's likely to be an antigen that can be used for making diagnostics, for discovering antibodies, for making vaccines like we did with Moderna. And because of the spike structure that was known, it informed all these other vaccine companies, and many of them use the stabilizing versions of this construct in their vaccine products. So over the last few years, Israel has probably been the most efficient or effective at deploying these vaccines, and they've estimated that even though they started vaccinating about a year into the epidemic, they reduced the number of infections by about 1/3, and they reduced the number of deaths by about 2/3. In this country, we've lost over 1 million people to this disease, and about half of them died before vaccines were generally available, and about half have died since vaccines were available. This is another thing that people have complained about and used as a cause for vaccine hesitancy. But if you look at the data from April of 2021, when vaccines were generally available, and ask who's dying from coronavirus, these are the data. And this has become everybody's favorite medical journal, the New York Times, and because they make the best graphics and the easiest to understand. But it's the unvaccinated group that are dying, and the people who've been vaccinated and still have severe disease are often have other complications, and many of them even have genetic polymorphisms in their type 1 interferon pathway. And so there's a lot of reasons behind why some of the vaccinated people are still getting sick. So during this pandemic, there's been 2 or 3 major narratives going on. We had primary health care workers who were doing heroic work, trying to keep people alive, working more hours, putting themselves at extreme risk, many of whom died. And yet, unexplainably, even as the good work they were doing, they often became targets of rage, even with death threats. Scientists were honored, but they were also threatened, and somehow we politicized biology, and we developed distrust of people in science and people with expertise. We are having a lot of physicians and nurses step away from primary health care. At least in many of the schools I'm familiar with, maybe not here, I hope, but medical school applications are down, they're not up. We have long COVID, and we're going to have a long tail of mental health issues to deal with over this next 10 or 20 years, and we've had an increase, not a decrease, in social disparities, including health care and access to health. So the question is, what are we going to do going forward? And there's several scientific opportunities. We have these existing technologies that I think we can be better prepared for future pandemics, and mRNA is a new technology that's going to be used for a lot of new things. There's still a lot of work to do. There's a lot of questions to ask, immunological questions that I'm listing here we won't go through. And there's still a lot of things to worry about. And so we can work on prioritizing these projects and try to be more ready for the next big thing, but I don't think we can be too selective because there's a lot of different ways that viruses can come back around to hurt us, like HIV that we were so surprised by because it had such a long incubation period. And so enteroviruses that cause polio-like disease. We don't know very much about bunyaviruses that could switch to respiratory spread and become more human-to-human spread instead of just vector-borne. Polyomaviruses or herpes viruses could have a long delayed effect that we may not recognize for a long time. And so all these things I think we need to think about and be more ready for. But what about just medicine in general? And we have a lot of opportunities. I'm listing some here, these technological capabilities we've talked about. We know a lot about immunology and immune correlates and the precedents that have been set during this pandemic for various product approaches and modalities and adjuvants and all sorts of things are going to change what we can do going forward. I think we also have a really strong proof of principle that basic science pays off. And, you know, just wanting to know what an F protein looks like somehow resulted in a better vaccine. And we also have somewhat of an obvious argument that we need improved public health infrastructure. We have lots of young people who've lived through this pandemic, and you would think that there is going to be increased awareness and interest in being viral immunologists, or at least interested in medicine and public health. So we should probably have thousands and thousands of budding vaccinologists out there, but we need to open the doors for them to be trained and have opportunities to do this kind of work. And I think there is a sense that we need better global cooperation and coordination. The challenges that we're facing are mostly, in my mind, social and political, and the misinformation, this politicization of biology, the distrust, and in those children who are maybe inclined to become vaccinologists, they've had their education disrupted. And many of our medical students who've been with us haven't been able to learn medicine in the same way that we had been used to learning medicine. It's been more virtual. So I'll wrap this up with some of these conclusions that we're in a new era of vaccinology. Combining precision antigen design and some of these platform approaches provides a more engineering approach to achieving more generalizable solutions for these problems. We have a lot of immunology to do. We have a lot of work still to do with innovation on mRNA. We're at the very infancy of RNA. There's a lot that young people could do in this area. And in my hopes for the future, we're going to have better global agreements, because if we can give access to the resources and reagents and technologies that can now be used to solve some of these problems and make them available to regional investigators around the world, and let these regional problems be solved locally before they become global problems. And I think there's easy arguments to make that there's mutual benefit to having this kind of distributive sharing going on. We have to have more consistency and diversity in our community education, because we have to build faith in science when we're not in a crisis mode. You cannot build trust in the middle of a crisis. And I'm hoping that as we go into this renaissance period post-pandemic, that's the narrative we want to promote, that we're going to have physicians and scientists who care more, and who care about the details, and who care about the patients, and care about the biology, and do it in a way with more compassion. Because you can't just do well, you have to also do good. And I'm going to stop there, thanking my lab that I've worked with these years, the VRC principal investigators, and especially Tracy Ruckworth and Masaru Kanakiyo are still there at the VRC maintaining the lab. Jason McClellan, who I've done most of this structure-based design work with over the years, he's a professor now at UT Austin. And I want to point out Dr. Kizmikia Corbett, who was a fellow in my lab during when this thing broke out and had been working on the coronavirus projects for 5 or 6 years before that, and was a big reason in those first 8 weeks why we got an mRNA vaccine from Moderna. So she's now an assistant professor at Harvard. And I want to thank you all for your attention and for my slides.
Okay, we've got some time for questions. There are some. So I saw that you're collaborating with Neil King, and I was going to ask you about the potential for using artificial intelligence approaches, AlphaFold, for protein structure prediction to get faster structural information, things like these surface proteins.
There's a lot of interest in artificial intelligence to solve structures. I wish Jason McClellan was here to talk to you about that. I'm not the right one to talk to you about it. But there is advances that, especially when you go in reverse, when you take a structure and then try to predict the linear amino acid sequence, that type of AI is, I think, gaining some traction. And there are sometimes there are some programs that can predict structure, but if you're making a vaccine, you really got to know what it is. And if they're only right 90% of the time, I don't think that's good enough to make a vaccine. And, it really has to be right. And I think if I've learned anything over these last few years, you've got to get it exactly right. Because if the antibodies aren't seeing the right thing, as Jim will tell you, it's just not going to work. And that's one of the problems with mRNA is that people see mRNA and they think it's the magic bullet and it's going to solve everything. But if the mRNA is not expressing the right thing, it's not going to work. So we really still have a long ways to go, I think, with antigen design before we can, see a linear sequence of amino acids and say this is its structure and that's the epitope.
Yes.
Barney, 1988, you're on the S corridor, and you're taking centrifuges out of the hallway that don't work and trying to repair them. Do you see, back then, could you see yourself here now doing what you've done over the last 35 years?
Well, Stokes, I believe in God's grace to just take you through some of that because you couldn't really have planned this. You couldn't see how the different threads come together at the right time, at the right moment, and being in the right place with the right kind of resources. So, I think you just have to keep doing the best work you can with what's in front of you. And I think Dr. Brittingham maybe made the best point of all. When I first came here as an intern, Dr. Brittingham, and there's a book written about Dr. Brittingham by Dr. Graber. Alan Graber wrote a book about TEB. You should all read that if you want to be good doctors. But Dr. Brittingham, I was in the library as an intern, which was probably not that common. And Dr. Brittingham came up behind me and snuck up behind me and tapped me on the shoulder and scared me to death. But he said, "Graham, this must be a great place because you're here and I'm here." and then he just walked away. But the point was, you can make it what you want it to be if you just keep paying attention to what's in front of you and taking step by step. And that list of people and names I put up there, the first medical student or the first fellow or the first postdoc who trusted me enough to be in the lab, or Dr. Oates, who let me go to graduate school because President Reagan pulled my physician scientist award back so that he could fund AIDS research in 1986, finally. Or, things like that, you just can't plan or predict. You just have to do the best you can with the situation you're put in, and then depend on God's grace.
Barney, I can't remember asking you when you were leaving, because I was so mad that you were leaving Vanderbilt. Asking you about it, and you said, I have to go because there's two things I want to do, a vaccine to HIV and a vaccine to RSV. So you may have taken a long time, but you were thinking about these things and planning it ahead, and that's how you went.
Well, I knew what I wanted the outcome to be, but I had no idea how to get there.
Yeah. But I want to ask you, what do we do for institutions? Think at the large level, but institutions to support young investigators. We do two things. One is to do fundamental discovery in biology and have the time and the bandwidth and the support to keep pursuing really fundamental questions that don't appear to one who knows fundamental questions that people don't think are very important for a long time. And then second, how do we create incentives that so that they're not institutions and young people aren't so risk averse because of all the concerns about working with pathogens?
I'm going to let you answer your own second question. But the first question is, I think Dr. Marnett has the hardest job in the room, because the trends right now in medicine and education are to be so focused on the finance and the business of all this that you forget about the patients and the biology and the training and learning. And it's taking longer and longer for people to grow up. And I feel like I was kind of a late bloomer. I never expected to ever even have a paper in science. I didn't have my first science paper until I was 60. And now I've had some science papers, but it took a long time to get that to happen. And the first R01 grants now are coming when people are well past 40. And it's not easy for leaders to have the patience with young people, and which young people do you invest in, and how do you do that, and how do you wait until they actually can be self-sustaining. This is becoming harder and harder because as the world gets faster, everybody's more impatient, but these things take time. So I don't know what the answer is, Mark, but the gain-of-function pathogen problem, I'm going to leave that one with you.
Oh, thank you.
I think one of the things that happens when you get old is that maybe you remember things perhaps in a little bit better light than they might have been. But I must say that it was really an amazing time when David Carson and Peter and you and Phil Gruber and
Phil Johnson.
I mean, it was so exciting to see the patients and see what these diseases did, and then to come back and sit around the table and try and understand the science and understand the pathogenesis. And no matter how hard I studied or how well prepared, I never had the answers. Carson always had a question that I couldn't answer that made me feel like I was inadequate and that I needed to study more. And it was a wonderful opportunity. It was a wonderful place. And I think that's what we all want. And that kind of excitement, has kept me here for 42 years. So I think that love of understanding disease and pathogenesis is a wonderful thing.
And sharing that across generations, because that's how it keeps building. So there have been unique places on Earth where things like, where things really coalesce and it becomes a great place for just discovery to happen. Like in Melbourne in Australia in the late 70s when Peter Doherty and Zinkernagel were there, and That's where Tom Bracciali trained, and you know, a whole bunch, the whole world was populated by immunologists who came out of that one place that did all that work that resulted in so much gain. And I feel like there was something like that at Vanderbilt. I felt like there was something like that at the VRC. The question is, how do you make that happen more often? And it takes strong leaders to hold back the business people while these young people are evolving. It's not easy, and it's getting harder, I think.
Hey, Barney, thank you very much. I think a lot of the best scientists are in love with what they do. I remember when you were chief resident at the General, you took a lot of pictures, many to show students and residents, many of which were infectious disease consequences. And, you know, if you're not in love with what you do, you probably don't do it quite as well. I guess what I'm going to ask you is, when did you discover you were in love with infectious diseases? Because that choice of viruses was almost unique for medical residents. And so just sort of talk about the way you fell in love with what you do.
Well, I've always enjoyed solving and fixing things. That's why I've said that my time on the farm as a teenager and young adult was where you had to spend about half the day fixing things or looking for things or trying to, you know, figure something out before you could do your work for the rest of the day. And there was always something to solve and always something to build, and you had to learn how to identify a problem, figure out a solution, and then actually do it and finish it. And I never really understood why they let me into Rice University until later when I was interviewing for Rice. And because, you know, I wasn't a typical Rice student. And I now know because the guy asked me how many books I read that summer, and I said, I didn't have time to read books. Well, what were you doing? I was building fence and building barns, blah, blah, blah. And what I was doing was identifying a problem and dealing with it and finishing it. And like we used to say in the lab, zipping the FedEx envelope and sending the grant or the paper off. You have to finish the FedEx envelope or hit the button to send the grant. You know, you can't just dabble and then go to the next thing. You have to finish things. And I think that's a lot what the farm taught me.
Okay, well, I'm actually not sure I had the hardest job in the room, but I think you made it a little easier because that was the greatest justification for basic science and its importance I've ever heard. So please join me in thanking Lloyd.